So, good evening, and welcome to Chapter 16, Sensory Motor and Integrative Systems for Bio 211, Norwalk Community College. Again, I'm going to say this every time I start making one of these. Even though I'm trying to help you hear an elongated sort of introduction to the material inside the textbook, remember there is no substitute for reading the book before you come to class and you're listening to the lecture. Obviously, I'd love you to watch these things and read the book so you can come in and participate in a conversation. But one thing I'm getting for feedback is people want more and more detail. Each of these things is supposed to point you in the direction of the detail you need to be paying attention to. I can put all the detail in here, but then I lose you. So what I want to make sure you're doing, watch these, read the textbook. You also have the Khan Academy things that I put up here for you. Make sure you are paying attention to what Mother Nature's doing here. And that is this question. How does Mom Nature create a system of pathways to collect and respond to stimuli? That's what you are. You have a series of responsive structures that send stimuli, these are neurons, that send stimuli into the central nervous system that will ascend, be interpreted in the gray matter of the cortex of the brain, if that's where it has to go, and will send some stimuli down to activate some motor response. And inside Chapter 16, we're literally thinking about what are the motor responses. How do you actually get muscular contraction to occur? So as we go through this, yes, inside the back of your mind, you've read Chapter 16, you understand that what we're going to do is we're going to put together a series of pathways, those tracks that you guys drew, and you want to know what types of information are carried on it. And what I don't mean there is the information, say, from Chapter 12, which was you have a depolarization event. It's assumed you understand that now. Now what you're doing is you're seeing which neurons synapse with which neurons to create what pathway. What pathway? And oops, that's not what you wanted to do there. Control Z. Let's get Control U, P. There we go. Okay, and those pathways ascend and descend. So let's get to work. Let's think about sensation first. Sensation comes in two forms. There's the conscious form. Go ahead, pinch yourself. And then there's a subconscious form, where you are aware of changes in the external and the internal environment. You may not think about it, but when you walk in a building that has air conditioning on, you may realize there's something different as a result of that. There's probably either less pressure inside that building, which draws more fluid out of your body, things like this. These are the subconscious awareness things. Or somebody blushes. You're visually stimulated, but you don't necessarily understand what it means. You will interpret these sensations. That's a bit outside of the purview of this class, and I'm already thinking I want to keep you focused on the types of pathways that we want to understand inside AMP1. We'll worry about the interesting stuff inside lecture with the questions that you guys ask. This stuff you understand. There's going to be some stimulation of our sensory receptor. Yay. We're going to transduce that stimulus. We're going to generate a nerve impulse. Notice now we're using the word generation and not depolarization. Okay. And what you see here is it's going to go to an integration point, and we'll think about what integration means in a second. General senses. For Chapter 16, we're thinking about somatic and visceral. We really tackle visceral senses when we're thinking about Chapter 15, but you want to be aware that somatic are going to be tactile, thermal, pain, proprioceptive. These are the things we're really going to be asking you questions about. But right now you're sitting there and maybe you're hungry, and I just mentioned the word hungry and now you feel hungry. You got that because you have a subconscious visceral ability to collect information that is now being interpreted inside your brain. Remember, we're going to use Chapter 17, smell, taste, vision, hearing, and equilibrium. A lot of this information travels back into the brain via cranial nerves, which means they have sort of their own pathway, designated pathways. Some of these pathways you understand if you think about, say, cranial nerve 1 versus cranial nerve 2. Okay? When we're thinking about sensory receptors, we're going to break them down into their types of sensory receptors. Okay? And here's where I want to introduce a specific type of thinking. So let's do this first. Let's go bold. Wham. Oops, that didn't go work out the way I expected it to. Let's do this. Bold. Okay. Your textbook changes its vocabulary right about here, and it says types of sensory receptors, and then it starts talking about modalities. So a modality is the type of sensation that you are conveying on a subset of nerves that are traveling to the cortex of the brain to be interpreted. The simplest, pain and thermoreceptors. These utilize free nerve endings, which means, for the purposes of exams maybe, thermoreceptors, changes in temperature, and pain. Somebody pinches you or you've been stabbed or something like that. They use free nerve endings. Remember the example I've given you in the past has been a paper cut. And then put salt water on the paper cut. Okay. Encapsulated nerve endings, these tend to be Pacinian corpuscles. Okay. Remember, there's a specific construction. It's a free nerve ending that is actually surrounded by connective tissue. Versus, say, separate hairs. Okay, where you actually have hair cells. Okay, those hair cells can be located in different locations inside the body. You're going to see inside chapter 17, you'll find them inside your ears. Photoreceptors, these are inside the retina of the eyes that you guys dissected. And the gustatory receptor cells. Remember inside class I said think of chocolate as it melts over your tongue. Okay? But you've seen this figure. We went over it inside class, but for those people who are not inside the class, I'm going to walk through this for you. This is going to be the activation of that first-order sensory neuron. First-order sensory neurons. So remember, inside this class, we have first-order activates, second-order activates, third-order neurons. So you need to know what type of neuron is being activated, meaning in this case, it's free nerve ending. And you can see here's our cell body. So we know it is a unipolar neuron that is activated to create a generator potential that will travel to activate over here. Let's go like this. We know over here now will be our second order neuron. Our second order neuron will ascend, it'll do something, and it'll activate a third order neuron. And I'll put the third order neuron over here, going over to the cortex of the brain. Okay, all the details will be in here, but here's number 1, here's number 2, and here's number 3. So unless you're told otherwise, all somatic pathways actually have 3 ordered neurons. You need to make sure you're learning as you're learning each of the pathways. Okay, but in this example here, you have a cold stimulus. It's traveling on a first order neuron. We cause a generator potential. It will propagate into the central nervous system. Likewise, first order neuron that's encapsulated. So here's the connective tissue surrounding that free nerve ending. Cause a generator potential. It travels into the central nervous system. The distinction would be a sensory receptor, specialized cell type. Here is a sugar molecule inside that milk chocolate that's melting on your tongue. Notice it's causing a receptor potential because it's a specific type of cell. It's releasing the neurotransmitter from a synaptic vesicle on that gustatory type cell here to activate now the dendrite of now that first order neuron, and that will then send signal into the central nervous system. So hopefully from these 3 examples, you can see that there's a pattern. There's a pattern is we have to activate a first order sensory neuron. Once we understand there's a pattern, how does Mother Nature now, okay, how does Mother Nature decorate the body so that you can actually activate all of these different receptors? So let's do this. Try not to mess this up too much. See what happens when I do this. Here's a really simple cartoon of you, right? You are a stick figure. You don't really look much like a bag of salt water right now, but you are, right? And that bag of salt water is a smiling face, right? And Mother Nature can decorate the outside of your body with dendrites that would be exteroreceptors. And then if you wanted to imagine inside of your body, she can detect different things by putting interoreceptors. So those are literally inside the body, facing inward, okay? And the dendrites of those receptors are facing inward, but they still collect the information. It still has to ascend to the brain. You still need to interpret it. I would probably collectively say that these are probably subconscious. Exteroreceptors, They may be subconscious until they become conscious. Like you don't think about the bug that lands on your arm until it bites you. You may not even feel it. Okay? The ones that are the hardest for people to wrap their brains around are these things that are called proprioceptors. And they're located throughout the body. And this always happens, right? I touch something and it gets way out of whack. So let's do this. Okay, get rid of that. Fantastic. Got rid of the cartoons. And now let's just figure out what proprioceptors are doing. Okay? Proprioceptors. Space of movement. Oops. What do I mean by that? I mean that you right now lift up your right arm, touch your thumb to your pinky finger, your middle finger, your index finger. Now go back to the third finger. And you could do that, right? And your brain can figure out where these things are. Close your eyes, take your index finger, place it on your nose. It is collecting a series of information from the joints and tendons inside the body so that the space of movement can be interpreted. So you know relatively where you need to place, say, a muscular output as a result of a sensory input. Another way to actually classify sensory receptors is this type of the stimulus. And earlier I mentioned things like modalities, and this is a great example. Not that the others were not, but you can actually have mechanoreceptors. Mechanoreceptors are actually the opening of force applied to a dendrite that was going to open up a channel. Thermoreceptors are obviously changes in temperature. Nociceptors, changes in pain. Okay. Photoreceptors, these are photons of light activating photoreceptors. Chemoreceptors, these literally are just detecting different chemicals. So you could imagine inside your nose you have chemoreceptors. On your tongue you have chemoreceptors. Food is nothing but a chemical. Okay. You may not think of it that way, but how does your body know if you have an acid or a base or a salt that's been placed on your tongue? She places chemoreceptors there. Osmoreceptors I've spoken about in the past, especially in class, making sure you pay attention to the fact that if you are a bag of salt water, and that is all you are, you need to figure out how much water is in that bag of salt water all the time. And the way to do that is to measure pressure. Okay? So the amount of fluid inside of a space exerts a specific amount of pressure. If it gets to be too high, you have to respond to it. If it gets to be too low, you have to respond to that. So Mother Nature decorates your body with a series of osmoreceptors to make sure you are performing properly. I won't even say optimally, but within normal homeostatic range. Now, not all sensory receptors behave the same. They can be rapidly adapting. Do this, go like this. Okay, and good examples of this are direct pressure, touch, and smell. And one thing I would say is, okay, put on a baseball cap. Okay, 5 minutes from now, do you still feel the baseball cap? Okay. Or somebody's cooking food when you're walking in the door and you're in the house 5 minutes later, do you still smell the food? I mean, all the chemicals are still there, but the receptors have rapidly adapted to the concentration of the odorant or the physical nature. Slowly adapting receptors are those that detect pain, body position, chemical composition of the blood itself. So chemoreceptors inside the aortic arch or the carotid bodies, they are not sending information, poorly informed information. Okay, your body, your life depends on making sure that they get that information correct. So having told you all of those things, we want to now sort of switch gears, but to do that I'm just going to simply stop this recording now with a little bit of an overview. What we've done is we've said that your body can collect and, whoops, come on Jonathan, collect specific types of stimuli. And what I want you to do is I want you to like mentally draw a three-dimensional image of yourself and then try and put different receptors where they could be. Okay? Remember that a generator potential is going to be created unless you're using a specific sensing cell, meaning, say, gustatory cell. So we have generator potentials versus receptor potentials. So G versus R. That's what I'm talking about, okay? And then you actually have rapidly adapting or slowly adapting receptors. So that would be R versus S, but the R's mean something slightly different here. We need to think about this because when we start putting somatic sensing tracts, it's these differences that determine what type of information travels to what location inside the thalamus that will ultimately go to what location in the cortex of the brain. And that's a pretty good place to stop. Okay?